Yeah, thank you, Adolfo. So, good afternoon, everyone. I'm Cecilia Picazo. I'm postdoc at the Department of Biochemistry and Molecular Biology at the University of Valencia in Spain. Today, we are going to talk about the topic ribosome biogenesis and regulation. As you know, ribosome biogenesis is regulated by several pathways. Let's see. This regulation, ribosome biogenesis, RIBI, is, as you know, is essential for different aspects of cellular homeostasis. In fact, cells invest an incredible amount of energy and resource into the ribosomes. And for that reason, this process is important for almost all the aspects of cell physiology and metabolisms. Okay. Moreover, the effective ribosome biogenesis has been linked to many human developmental disorders, as you know, as cancer, initiation, and proliferation. So this process involves the 3 RNA polymerase, and it is really well regulated. I'm not going to talk more about this. It's only to do a short introduction. And as you know too, there is a huge pathway that regulates this biogenesis, that is the mTORC1, okay, that is a nutrient sensing pathway. So in this context of ribosome biogenesis and the regulation of this, Ferhat Alkan from Ribosome Dynamics Lab, division of Oncogenomics, in the Netherlands Cancer Institute in Amsterdam, will talk about ribosome dynamics and a new tool, a bioinformatic pipeline, ARF, which integrates ribosome RNA fragment data with the 3D structure of the ribosome to predict change in ribosome heterogeneity across samples. So now I'm going to stop to share, and now it's your turn, Ferhat. Thank you so much.
Thank you for the introduction. I'll quickly, like Cecilia mentioned, I'll be talking about our tool to predict ribosome heterogeneity and ribosome collisions from the ribosome profiling, so ribosomal RNA waste data. And yeah, I call it trash or treasure because we are basically trying to turn the ribosomal RNA read trash of the ribosome profiling into a beautiful bioinformatic treasure where we predict the heterogeneity and the collisions of ribosomes. So to start the topic, oh, my slide is not changing. Yes. So the ribosomes, as you all know, are responsible for the RNA translation process in the cell. And they are very complicated machines, and they have different roles in different parts of the whole translation process, in the initiation, elongation, termination, recycling. And the ribosome is this beautiful machine which can coordinate all these parts of the translation process. And this is something you all know, but maybe something you don't know is that the ribosomes are actually very, very abundant. It's the most abundant macromolecular structure in the cells. And around like 70–80% of the cell volume is actually taken by the ribosomes itself. And also, like Cecilia said before, that 70–80% of the all energy produced in the cells are spent by the ribosomes for the RNA translation process. And if you look actually more deeper into this ribosome structure itself, you also probably, some of you already know that that the ribosomes are made from these different ribosomal proteins, around 80 different ribosomal proteins, and four different ribosomal RNAs. And these ribosomal RNAs are 5S RNA, 28S, 5.8S, and I think in the next talk we will also see some different features of this 5.8S RNA, ribosomal RNA, and the other one is the 18S ribosomal RNA. And we say that there are around 80 different ribosomal proteins inside the ribosome because this is not a very there's no definite answer to how many of them are actually inside the ribosome, because we have this beautiful macromolecular complex structure of the ribosome, and then because of this whole complexity, we know that there is also heterogeneity inside the ribosomes. So meaning that inside the cell, when you look at different ribosomes that are part of the cells, you can actually have different heterogeneity coming from different parts of the ribosomes. For example, from the rRNA point of view, you can have different ribosomal RNA variants inside the ribosome. And the ribosomal RNAs also can also be modified differentially between different ribosomes inside the cell. And also from the aspect of the ribosomal proteins, we can have different stoichiometry of the ribosomal proteins inside the ribosomes, meaning that some ribosomal proteins are actually not always necessary for forming the whole ribosome, for forming a functional ribosome. And sometimes these ribosomal proteins can be in the ribosome through their paralogs, or sometimes they are not just there at all. And sometimes the ribosomal proteins can be also in different parts of the ribosome, and there can be different modifications also on the ribosomal proteins, meaning that at the end, there's a big heterogeneity inside the cells when it comes to what kind of ribosomes they do have. And for example, from the ribosomal protein point of view, in relation to cancer, we now also know that there's a lot of different mechanisms that are actually regulated through this ribosomal protein heterogeneity of the ribosome. So when the cells are between cancer or normal cells, they have different ribosome populations. They regulate their population of the ribosomes, and through the stoichiometry of different ribosomal proteins, and then this regulation leads to a further regulation in the different hallmarks of the cancer, in the cancer research, in different hallmarks of the different regulations inside the cell. But these are not only sources for different heterogeneity. So like I said before, the heterogeneity of ribosomes can come from the different ribosomal RNA variants, different ribosomal proteins, but also the heterogeneity of ribosomes can also come from other aspects. For example, there can be different proteins that are associated with ribosomes in the cell, or ribosomes can be in different parts of the cells. They can localize to different areas inside the cell. And the ribosomes can also be in the cell in different architectures. For example, a very nice example for this is that there can be ribosome collisions, for example, inside the cell. Meaning that sometimes the ribosomes during the translation process can come very close together. For example, if there is some kind of stalling due to amino acid depletion, or due to resources, or due to some other reasons like faulty mRNAs, or any other, for example, different agents that stall the ribosomes. So when this ribosome stalling happens for a really long time, the ribosomes can come and collide with each other, forming then this disome structure that we call them, or sometimes the trisome or quadrosome, depending on how many ribosomes are collided to each other. And this is very important because recently in the last decade, we discovered many new mechanisms, how these different kind of ribosome collisions or ribosome stallings are recognized through the different sensor proteins. For example, ZEC alpha is one of them, GCN2 is one of them, DNF598 is one of them. And then all these different proteins that senses these ribosome collisions leads to different responses inside the cell. For example, through ZEC alpha, you can have ribosomal stress response. Through GCN2, you can activate the integrated stress response, or through ZNF598, you can have the ribosome quality control machinery kicking in. So we have recently started seeing more and more cases of these ribosome collisions inside the cell. And then when these ribosome collisions happens, we see that then this creates a cascade of events which sometimes change the behavior of the cells, or it even, for example, we had a recent paper where we showed that, for example, in the intestine, adult stem cells turn into fetal stem cells. So there are many different biology is explained through this ribosome collision. So it's also very important heterogeneity inside the cell. So it's nice to know if there's more collisions or less collisions in certain conditions. And also, if you look at the PubMed recently, you see that ribosome collision is actually getting picked up by more and more researchers, and then there's new mechanisms and new discoveries being made. But when it comes to detecting these parts, this different heterogeneity of ribosomes, whether it's ribosome collisions or it is heterogeneity from ribosomal RNA or from the ribosomal proteins, how do we detect them? Because this is a very new topic and new area, and it actually requires a lot of experimental efforts to recognize each kind of heterogeneity that you see here. For example, for the ribosomal RNA part, you can do different sequencing protocols to detect the variance or modification differences between two conditions. And for the ribosomal protein part, you can do different mass spec-based approaches to measure the ribosomal protein abundance inside the active ribosomes. And the heterogeneity from other aspects, for example, the ribosome collisions, there is some very complicated approaches, like indirect approaches to measure how much collision is happening between one condition compared to other. But these are all experimental efforts. But to change this, to advance this field a little bit further, what we did recently is we also focused on the heterogeneity of ribosomal proteins. And then we showed that we recently published this tool that by using the ribosome profiling data, you can actually predict the ribosomal protein heterogeneity between your conditions. And this is really nice because this means that if you have such tool that uses this ribosome profiling data, you can also now go back to all the studies that are out there that has done ribosome profiling and reanalyze these data sets from the ribosome heterogeneity point of view. And you can have some candidates, ribosome heterogeneity candidates for explaining different biological phenomena. But how did we do that? Like, how did we develop this tool? We did this through our observations of this whole riboseq protocol. So as you all know, that the riboseq protocol works in the sense that you first isolate the ribosomes from your cells, and while the mRNAs are still attached to it, where the ribosomes has stalled on this mRNAs, and then after that you use this nucleus digestion, where we treat them with RNases, where we cleave out the mRNA parts that are not protected by the ribosomes, so that we only end up with these fragments, small mRNA fragments, that are protected by the ribosomes itself. And then by using these reads, we can quantify the translation dynamics in our cells between different conditions. But the one thing we notice here is actually this nucleus digestion doesn't only work on the mRNA sequence, because there is also the ribosomal RNA inside the ribosome. So what happens is actually when you digest, when you do this nucleus digestion, when you do this RNase treatment on your cells, you also cut the RNA, ribosomal RNA on the ribosome from at different parts of the ribosome. So it's not just the mRNA that is being cleaved, also the ribosomal RNA is being cleaved at different parts of the ribosome. And because of that, after that, when you try to sequence all these fragments coming from your nucleus digestion, you end up with not only mRNA fragments, but also these ribosomal RNA fragments that are coming from different parts of the ribosome. And when you start looking, we usually try to deplete these ribosomal RNA fragments, thinking that this is actually a noise because there's no information there. But then if you start thinking about these ribosomal RNA fragments from the ribosome heterogeneity point of view, you can clearly see that these ribosomal RNA fragments can have some information about the structure of the ribosome. Because when you compare a standard ribosome to another ribosome, which is, for example, missing this one specific ribosomal protein, when you do your RNase digestion protocols inside the ribosome profiling, you can assume, you can see that, you can think that this region that is protected by this ribosomal protein would actually be digested differentially, since this protein is not protecting this ribosomal RNA fragments in this region anymore. Because this region, because of the ribosomal protein is not being there, would be more exposed to maybe RNase digestion at different parts. So at the end, when you get your ribosomal RNA sequences from your ribosome profiling data, you can assume that there will be some differences in the rRNA fragments that you are sequencing that are coming from this region where this ribosomal protein is not there anymore. But this is just the way that we were thinking that maybe this might be the source of the heterogeneity of the RNA fragments in the riboseq data. But in the normal case where we performed our riboseq data, we don't really know which ribosomal protein could be a candidate for this heterogeneity between these ribosomes. But what we know is we just know that there are differences between the ribosomal RNA reads between these two different conditions where we performed our ribosome profiling. So what you have to do in this sense is that you have to somehow develop a reverse engineering approach in here, where you try to make meaning out of all these fragments and how they are related to the different ribosome population in the cell. So which ribosomal proteins are, for example, is missing from the ribosome, or is there in one condition more than the other. If there's any questions around like here, you can always stop me and ask the question if you. So how did we do this reverse engineering approach in our tool is, of course, we first needed to understand which ribosomal protein protects actually which ribosomal RNA fragment. So to do that, what we did is we analyzed the 3D structure of the ribosome. This was done with the human ribosome structure. And what we did with this analysis of this 3D structure is we calculated basically the distance between every ribosomal protein to every rRNA position. So in this plot, what you see here is that this is the ribosomal RNA sequences that are inside the ribosome. This is the 18S, 28S. These are the 5.8S and 5S ribosomal RNA. So in each line here, in each column that you see a ribosomal RNA position. And here on the y-axis, what you see is that you see a different ribosomal protein. So what we measure in this 3D structure is that which parts of the rRNA is in contact with which ribosomal proteins. So if you look at each row, you can see that each row is slightly different from each other, meaning that each ribosomal protein is in contact with different parts of the ribosomal RNA. Meaning that each ribosomal protein is protecting a different part of the rRNA from the RNase digestion. So by using all this information about the ribosomal protein rRNA proximity map, what we did afterwards is we developed our pipeline. So in our pipeline, what we do is we get the riboseq data from two different groups, and we separate our rRNA fragments from this riboseq data. And then what we do is we do a differential ribosomal RNA fragment analysis at each rRNA position. So we figure out which are the regions that are changing in the rRNA fragments in this riboseq data set between the two conditions. And then by using this information about the proximity of ribosomal proteins to ribosomal RNAs, we do certain enrichment tests. So trying to find out if certain RNA fragment differences are coming from a specific region that is associated with a certain ribosomal protein. Because, for example, if there is a heterogeneity of ES25 base heterogeneity, for example, if one condition you have more ES25 containing ribosomes, but in the other one you have less, we assume that there should be a fragment changes enriched in the region where ES25 is in contact with the ribosomal RNAs. So our pipeline at the end, by doing all this complicated analysis, it creates these candidate ribosomal proteins for differential ribosome heterogeneity between two groups. And of course, we benchmark this approach using the most famous ribosome heterogeneity data set from Maria Barna's group, where they created this riboseq data for all ribosomes and US25 enriched ribosomes, or UL1 enriched ribosomes, and all ribosomes in mouse embryonic stem cells. And when we run our prediction between these two ribosome populations, for example, you could indeed see that ES25, for example, was one of our top candidates in this comparison, where you expect to see that ES25 is indeed the biggest heterogeneity candidate for you. And in these plots that you will also see is that if something is on the top right, so around here, these are our best predictions, and these are the least important parts. So these are the ones that are predicted not to be heterogeneous, and these are the ones that are being heterogeneous candidates. And it was the same when we did the UL1 enriched ribosome, the ribosome population riboseq compared to the all ribosomes. You could also see that UL1 is one of our top candidates in this comparison. In our paper, we also had done a few other benchmarking comparisons, and we could see that indeed the tool itself works when we know that there is a riboseq data from a heterogeneous ribosome population, and we could indeed predict that these are the ribosome proteins that are candidates for this heterogeneity. And then after here, we didn't stop. Then we started also asking ourselves, what about ribosome collisions? Because when you think about ribosome collisions, where two ribosomes are sitting next to each other, you can also imagine that if there are more collisions in one condition compared to other condition, there should be also some changes in the ribosomal RNA fragments coming from the riboseq data. Because, for example, this region that is in the collision interface maybe should protect certain ribosomal RNA fragments more than the rest. So we started thinking about this also collision prediction. But then we noticed one thing, because defining the collision interface is one thing. Yes, this collision interface we can easily define, but actually defining which rRNA positions that would be protected from RNase digestion is another thing. Because when your ribosomes are in a collided form, when they are in this disome formation, it's not just the ribosome collision interface that is actually changing, because both ribosomes in this collision are actually having a slightly different structures when you look at the 3D structures of ribosome collisions. Because also, this is also because the ribosome collisions is not only in the disome structure, sometimes they can also be in the trisome structure, for example, and then this would cause differences in RNAs digested in different parts of the ribosome. It's not just this interface, it's actually because the structure of this one is different than this one. There will also be some differences in other parts of the ribosome for RNAs digestion as well. So what we did to find out what are the regions that are changing in RNAs digestion is we used the solvent accessibility approach. And the solvent accessibility is basically finding out which parts of the 3D structure is more exposed to the external solvent. And you can also think it as a way that the solvent in our case is an RNase. So it's this RNase is going all around this 3D structure of the disome, and then it's trying to access different parts of the rRNA. And by using this approach, you can somehow model the accessibility of different rRNA positions to the solvent or to the RNase in the case of ribosome. And how did we do this? We did it based on, basically, we took 4 different collision structures from the protein data bank, and then we analyzed them from the solvent accessibility on all these different structures. And then what we did is we compared the accessibility of rRNA positions to the solvent in the leading ribosome compared to the trailing ribosome. So we tried to find out which parts of the ribosomal RNA is differentially accessible to the RNase in the first ribosome or in the second ribosome, because if we can find these differences in the positions where we have this difference, it means that these parts are the ones that are reacting differently to the RNase treatment in the riboseq protocol. So by analyzing all these 4 different structures, we created this structure-specific collision sets, so 4 different collision sets. And when we start visualizing it, we also saw that some of these positions are actually in the interface itself, the red ones and the blue ones. But also there are some other parts that are actually probably being differentially digested by RNase, slightly because of the structural differences between this leading and the trailing ribosome. So how did this help us, like creating this set? This helped us in the way that we can finally create a certain collision set, which would show ribosomal RNA fragment differences in the riboseq data, and simply because of the differential accessibility, and in the case of if there are collision differences between these 2 comparisons. So what we did is basically after creating the set, we fed this into our differential RNA fragment analysis pipeline, this ARF pipeline. And by using this different set, which is similar to the sets that we created for the ribosomal proteins, we can now do this nice analysis of that. Next to the ribosome heterogeneity predictions, we can also now finally predict if there are differences in the ribosome collisions between 2 conditions. Of course, we had to benchmark our approach. And what we did is, for example, we took this data set from Yeast study, where they performed this ribosome profiling for different treatments of yeast cells, where they know that, for example, anisomycin or 3-AT treatment induces ribosome collisions. And we can beautifully see that in these comparisons, for example, control versus anisomycin, ribosome collision prediction was our first prediction in terms of the enrichment that we were doing. So we could indeed see that the ribosome collision is predicted to be different between these 2 samples, where the one group is treated with anisomycin compared to the control. And this is indeed what is expected, because we already know that the anisomycin induces collisions in these yeast cells. And it was the same when we look at the 3-AT treatment, which depletes the histidines in the cell. So it causes also ribosome collisions. And indeed, the ribosome collision had a very strong prediction. So this line that you see here, the dashed line, is our threshold that we use. Anything to the right of it is a strong candidate for ribosome, for enrichment of collisions or enrichment of ribosome heterogeneity. And when we look at some control comparisons in this data set, where for example, 10 minutes or 60 minutes treatment, or control versus cyclohexamide, this is a control comparison because after the cyclohexamide treatment, there is no waiting. So it's just instantly cyclohexamide treatment without any waiting for collision. So of course,
in such case, we don't expect any collisions because there's no time to create collisions. And indeed, in this control comparisons, we didn't predict the collisions on the other side. And we also used another study. For example, in this study, what they did is they did a riboseq in IP samples for HAL2 is a ZNF598 homolog in yeast, and it's known to bind to ribosomes when they are collided. So when you do a ribosome profiling in a IP HAL2 samples compared to the normal riboseq samples, of course, in one condition you have all HAL2 bound disomes, HAL2 bound collisions, and you do riboseq on them, and then compared to all the other riboseq, of course, you expect differences in the ribosome collisions between the two. And indeed, when we compare this HAL2 IP versus background ribosome collision, background ribosome profiling, we could indeed see that ribosome collisions are one of our top predictions by our tool. And also here in this comparison, it's done in the yeast cells where the SCI2 knockout combined with DOM34 knockout, which is known to resolve the collisions. So in this comparison, you expect that there is actually difference in collisions because of the DOM34 knockout. So because of that, we also know that there's collisions in this comparison, and we can indeed predict this as well. But this was not the all benchmark data because we also collected a few other studies from out there that is out there. And in all these comparisons that we know where there is an increase in collisions, we could indeed, when we do our predictions with this enrichment score 2 that I was looking at, which is like where we use this threshold, we could indeed see that almost all of our predictions were above this threshold, which means that there is a prediction of ribosome collisions in these comparisons. And in all the controls that we selected, you could see that our prediction scores were much lower than 1. It was around 0. So it was in the background level. So also when you compare this ribosome collision prediction with the prediction of ribosome heterogeneity of all the different ribosome proteins, you see the clear difference that ribosome collisions is predicted with much more confidence in these comparisons. And another thing here that I show is the collision interface. So if we had defined our collision prediction set only from the collision interface itself, so by just looking at the rRNA fragments in this region, this would be our prediction score. You can see that here our approach that we used with the solvent accessibility is much more superior compared to the defining the collision interface. So from our prediction point of view, it shows that we actually followed a really good approach and better approach than just looking at the collision interface. And as you have also seen in the previous slides, that I showed that there's also this ribosome heterogeneity prediction that we perform next to the ribosome collision prediction. And then so we look deeper also into this data, into this prediction scores across all these benchmark comparisons that we have defined before. And what we also have seen there is that when we predict ribosome collisions in these benchmark comparisons, we were also sometimes predicting a heterogeneity of certain ribosomal proteins between these two conditions. And US17, ES4, ES7, and ES8 was the strongest ones in these comparisons. And you can see that they're also usually high above for example, ES17 is high above our threshold line, also for ES7. This is also interesting because we also know that ES7 is known to be ubiquitinated when the ribosomes are stalled. So indeed, there might be some interplay that is happening when the collisions are happening. The ES7 ubiquitination is also happening. Or maybe there are some also unexplored mechanisms for US17 or ES4 in the cell when there is more or less collisions. And this is something we also want to follow up later. So in summary, what I showed you is now for any organism, you can perform ribosome heterogeneity and ribosome collision predictions using our RF package on your riboseq data. And we also show that there might be a potential interplay between this ribosome heterogeneity and ribosome collisions phenomena. And because of this consistent prediction of collisions with US17, ES4, ES7, and ES8 ribosomal proteins. And we are also now currently performing large-scale assessment of ribosome heterogeneity and collision predictions on human and mouse riboseq datasets. We collected around 170 different datasets where we are using both tools to predict different kinds of heterogeneity or collisions changes. And stay tuned for this part of the project in the next months that I hope we will have some results. So with that, I would like to thank everyone in my lab, especially William Faller, who is our principal, who is our PI, and giving me this opportunity. Edwin, who is also collaborating with me on this project, and all the other members of the Ribosome Dynamics Lab, and our collaborators in CRIC and here in NKI and the department and all the facilities. And thank you for listening. I'm happy to answer questions.
Thank you so much. So if someone has some question, it's time maybe for, I don't know, 2 questions.
Mm-hmm.
Hello, can I ask a question?
Yeah.
Of course, first of all, congratulations, very nice presentation, very clear, your data, very interesting. I have, let's say, a couple of questions. One is regarding the proximity map that you developed with ribosomal proteins. Would it be possible also with the modifications that we know were placed in ribosomal RNA?
Mm-hmm.
Do you have a project about this?
Yeah, we also discussed this, that if there is differences in the ribosomal RNA modifications, it would not cause any differences in the RNase treatment. But what might happen is that it might have differences in the sequencing. So, for example, if there's a pseudouridylation, there would be differences in the sequencing. And you can actually use this. Actually, there's already a paper who showed that you can use this normal RNA sequencing to find the pseudouridylation sites. But when it comes to the other modifications, if we can define what changes in the RNA sequencing for these RNA fragments, then we can also, of course, look at it. But I don't think, other than the pseudouridylation, we don't really know which other modifications change what in the sequencing. So if we can define this, then we can also look at this ribosomal RNA fragments coming from RiboSeq from that point of view as well.
But in principle, I think that also you can expect some structural modification if the modification is present or not. So maybe
you can also define a RNA position set around that modification site, and then you can also try to see if there's differences in the fragments only in this region. You can try it that way as well, but it would be a small site, because when you look at the ribosomal proteins, they are big and there are multiple sites. When in the modification of ribosomal RNA, it's just one small area.
Okay, thank you very much.
Thank you.
Can I ask a question?
Yes, please.
To me?
What's the advantage for the collision study? By the way, really nice tool. But for the collision part, what's the advantage of looking at the rRNA accessibility rather than the mRNA accessibility? What I mean is that with disome and trisome, you expect to have a difference in length of the RPF. So you would expect that if you have collision, you have higher amounts of longer reads compared to
that's already out there, like where people do disome sequencing, trisome sequencing, and try to find out where they are. But what is beautiful about this tool is that you just do a normal ribosome profiling. We don't provide a new experimental method. So what we provide here is that you just do your ribosome profiling to explain your translatomics differences, and then by using this tool, you can also have an additional ribosome collision prediction. So it's for us to create a new direction based on an existing data that you already have. And because in the ribosome profiling, you only select the region around the 30 nucleotide, and you don't look at all the other ones as well. But if you have a larger sequencing window, then actually our predictions probably gets even better, because then ribosomal RNA fragments come on all different sizes. They are not only in 30 or 50.
Thank you.
Maybe I have one more question. Have you been analyzing also the size of footprints? I mean, heterogeneity of size of footprints, because usually on the mRNA, yes, usually the classical footprint after ribo sequencing, it is around 28 nucleotides.
Yeah.
Yes. Have you been looking also at the different size, maybe regarding ribosome collision or something like that?
So in the normal ribosome profiling protocol, we usually, almost everyone always look at the specific size window so that they can get the standard RPFs around the 30 nucleotide. But then also people also sometimes get to 21 nucleotide, which is like a shorter formation, like a other formation of the ribosome. And people also show that actually when you analyze these two fragments and their ratio to each other, you can sometimes also predict the stalling. But when it comes to predicting the collisions, you have to sequence not only the 30, but also the 60 nucleotide or 50 nucleotide. But in the normal standard ribosome profiling protocol, you exclude those parts. You don't actually include them. But if you actually include both of them at the same time, then you can actually do a better comparison, like 50 nucleotide, 60 nucleotide ones compared to 30. Then you can have a better estimation of collisions. That's also true. But the standard ribosome profiling is usually limited around 35 nucleotide.
Okay, thanks.
Because it also, when you have a larger window, it increases the ribosomal RNA fragments that you're going to have.
Great. Thank you so much for your talk.
Thank you very much.
Yeah. And now it's time to Giulia, that she is from Department of Medical and Surgical Science at the University of Bologna, Italy. And she's going to talk about 5.8S ribosomal RNA isoforms and ribosome heterogeneity. So yeah, Giulia, when you want, you can start.
Okay, let me show the presentation. Can you hear me?
Yes.
Okay. Okay. So we thanks for the presentation, and thanks for the possibility to give this this presentation about my data. As we heard from the previous presentation, ribosome heterogeneity can originate from many sources. For example, the incorporation or the modification of ribosomal proteins, or the sequence or the modification of ribosomal RNA. And this heterogeneity will lead to ribosome specialization that can, these particular ribosomes can translate particular mRNAs, and this is true in particular for cancer cells in which oncoribosome can sustain the tumor growth. Ribosomal RNA 5.8S can be considered a source of ribosome heterogeneity, so far not considered, because of the presence of the history isoforms. 5.8S rRNA is positioned in the large subunit of the ribosome and plays a role in polypeptide elongation and mainly in the process of translocation. It has been studied particularly in yeast, in which two isoforms have been reported, the short and the long form, while in mammalian cells, recent publication report also the presence of a shorter isoform, which is called the Croft. And as you can see here from the sequence of human 5.8S, we have the starting point of the short isoform, which is the most abundant in the cell. Then the long form present 5 nucleotide more at the 5', while the cropped isoform present 10 nucleotide less from the short form, so therefore is considered also the shorter isoform. Little is known about these isoforms, and because mainly they are less present compared to the short form. In fact, crystallographic structures reported so far contain only the short isoform. And therefore, there is no characterization of the role of these different isoforms, either in cell biology and or in human pathology. And another question that we asked to ourselves is if this isoform can be modulated, and in this way we can have models to study their role in the ribosome and in cell biology. And I will try to give some answer to these questions with this presentation. Ribosomal RNA 5.8S derives from the 47S transcript that contains also the sequences for 18S and 28S ribosomal RNA. And it starts in particular by the processing in the internal transcribe space that is positioned between the 18S and the 5.8S sequences. Mainly by the cut in this position in site 2 by the RNase MRP complex composed of the long non-coding RNA RMRP and the 10 associated proteins. And this RNA, the 32.5S, can be either trimmed by XRN2 or cut in this particular point, putatively in this point B1, by an unknown enzyme to give rise to the long isoform. The trimming of XRN2 can stop at the start of the short isoform if the protein RPL17 is present, and also, or stop at the starting of the cropped isoform if RPL17 is not present. This is a recent publication and by a group of Wang in 2015. Then the 3' of the sequence is processed the same for all the 3 isoforms by the cut of last one and the trimming of other isonuclease to give rise to the 3 isoforms. To have a good image of these 3 isoforms in total RNA and to quantify them also in difficult samples that are not good enough to perform northern blotting analysis, we optimized a technique, a very simple technique based on primer extension coupled with fragment analysis. And in this technique, we perform a primer extension with a reverse primer conjugated with a fluorophore that allows the possibility to reverse transcribe the whole sequence of the 3 isoforms. Then this cDNA is subjected to capillary electrophoresis and fragment analysis. And as we can see from the electropherogram, it is really easy to distinguish the 3 isoforms based on their size. So here we can see the peak for the cropped isoform, the peak for the short and for the long. And by quantifying the area under the peak and normalizing by the total area of the 3 peaks, we can express the percentage of presence of each single isoform. Of course, there are new techniques like ribosome, like Oxford nanopore technique that can give the sequence of the whole transcript, but if I understood correctly, it is not possible to sequence the 5' nucleotides. So in this case, it would be not possible to distinguish the 3 isoforms. So to conclude, we can say that all the 3 isoforms of 5.8S rRNA are included in actively translating ribosomes, although with some differences between the cell lines. That we observe a modulation of the distribution of the 3 isoforms in a mouse model for cancer development. And we confirmed this in a breast carcinoma series in which we observed the correlation with tumor grade and estrogen receptor status. And also we confirmed the link with the estrogen receptor status in a cellular model. And also in analyzing the survival of these patients. And at least we managed to modulate one of the three isoforms, the cropped isoform, by knocking down RPL17. For the future, we would like to expand the ways in which we can modulate these three isoforms, for example, by using the data presented by the group of La Fontaine, in which they silence different factors involved in ribosomal RNA processing. And for example, these two factors seems to impact on the production of the long isoform. And of course, to obtain this model will be very interesting to characterize the structure, the 3D structure of these ribosomes containing different the different isoforms of 5.8S, and also study the efficiency of the ribosome or the capacity of the ribosomes to translate particular transcript. With this, I conclude. I would like to thank all the group of Professor Montanaro and Professor Penzo for the helpful discussion about this topic. Our funding partners, in particular IRC, and our collaborator here at Bologna. And thank you for your attention. I'm open to questions if you have ones.
Thank you so much. Nice presentation. So now it's time for questions. Someone?
Hello, thank you for your nice presentation. I was wondering whether you could separate the ribosome population containing one or the other isoform, and then eventually use them in vitro translation systems. Or
for now, we didn't find any way to separate them, to distinguish them. The only way is to force the expression of one particular isoform among the others. What I obtained with the cropped isoform is more or less, I think, what we can it is a starting point. Maybe we can increase the presence of one of the isoforms compared to the other, but for now we couldn't find any way to select them.
Thank you. Very nice presentation.
Thank you.
So one maybe other question. My colleague next to me, Hichem, was asking whether maybe you can see this modification during development or in.
It is indeed a very interesting question, and we will for sure look for that also in development of the human different tissues. It is quite interesting to see if there is a moment in which there is one or the other isoform that takes advantage, but for now we didn't look at that.
Thank you very much.
Thank you.
More questions? So if we don't have more questions, I think we're done here. So thank you so much, both of you, for the nice, really nice presentation. And see you in September, because now we don't have more webinars. So in September we will see again.
Thank you.
Thank you.
Thank you.
Bye.
Thank you.
Bye.
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 Thank you. Bye.
Bye.